So now it's my pleasure to introduce our first speaker. Professor Zhenping Qin is an assistant professor of mechanical engineering at the University of Texas at Dallas. He is a founding member of the Center for Advanced Pain Studies and holds adjunct appointments in bioengineering and at the University of Texas Southwestern Medical Center. Professor Qin's lab focuses on developing new nanotechnologies to better understand and treat diseases in the nervous system and to provide highly sensitive and multiplexed methods for infectious diseases testing. Dr. Qin has received numerous awards, including an NIH award for his pioneering work on the development of molecular hypothermia for optical control of protein activity. Today, he will speak to us on probing molecular and barrier activity in the brain.
So today I would like to share with the audience on some of the work we're doing in using nanotechnology, nanomaterials in the brain, and in combination with photonics and light. So I hope you find this interesting, and we'll look forward to some interesting discussions. So first of all, I would like to acknowledge the folks that has worked very hard in the lab doing all the work, developing a lot of this I'll be talking about. I'd like to acknowledge our collaborators, both developed at Dallas and also around the U.S. and sometimes in Europe and other countries. And I would like to acknowledge our funding resources from AIH and SAF, American Heart, CIPRATE, and DoD. So to get started, the brain is it's considered the most complex organ in the body. So to give you some numbers, so it's about 2% of body weight, so about a couple pounds, but it uses 10 times more energy and oxygen. So it uses around 20% of the energy and oxygen to support all the activities and processes in the brain. So there are about estimated to be 100 billion neurons, and each neuron connects to 5,000 other neurons, so that makes up about 500 trillion synaptic connections. So the brain can go wrong, the nervous system can go wrong. There's a lot of diseases that are increasing because of the age, society aging. So the Alzheimer's, brain cancer, stroke, traumatic brain injury, chronic pain, and so on. So over the last few years, the field has come to a consensus. One of them is that we really need new tools to understand and treat brain diseases. So, one of the tools that comes, with I have some experience is the nanomaterials, nanomedicine, nanoparticles, and with the work over the past 10 years, 20 years, we can now make particles, nanoparticles of all kinds of size and shape and composition. And a lot of them are gold, so this is referred to as the golden age of the nanomedicine development. If we look at the scales in terms of the the processes, in the brain, so we have the whole brain that's about a human brain is a lot bigger, but a mouse brain around centimeter. You have the circuit, and it's on the millimeter scale, and you go down to neurons and processes and vesicles and proteins. So this is a multi-scale problem. How does this interface with the nano, right? So nanomaterials interfaces at the around tens of nanometer, hundreds of nanometer range. You can see this would interface very nicely with the molecules. Can mimic the vesicles and and so on. So this will be kind of the focus of the work today. We also do some work in the virus diagnostics. So you can see the nano particles, nanomaterials. It would interface nicely with the virus particles as well. So just as a brief introduction to what we do, so we at the interface of the nanomaterials, photonics, and looking into applications in to try to make an impact in the brain and a virus diagnostics. So specifically, we're looking into develop new tools such as molecular hypothermia. I'll be talking to you about how to cross the blood-brain barrier, which is a major barrier for drug delivery into the brain. How to understand the fundamental processes in the brain with the tools, the nanomaterial tools. And lastly, how can we make a faster, better, multiplexed virus diagnostic tools? As everybody knows, everybody's suffering from the impact of COVID. So this becomes particularly important. So for today's talk, I would like to focus on in the brain area, try to tell you 3 stories in terms of how to manipulate protein activity using a new tool, what we call molecular hypothermia, how to build tools to understand the basic processes in the brain. I'll explain to you what the brain Wi-Fi is and how to cross the blood-brain barrier. So let's get started with the first topic. So if you read about the area of neuroscience or biology, biological science in general, or medicine, and you find this tool called optogenetics. So this is a method that uses optical stimulation of genetically transfected neurons. Basically, you transfect neuron with proteins, membrane proteins that can respond to light. So you can use light to excite or inhibit neuron activity. So this is considered a major breakthrough in the past decade. It took about 10 years to get into the clinical trials. The major challenge of the optogenetics is in the name. So the first is optics. So how to get light into the right place. This can be challenging in the brain. It can be much easier in other areas, such as in the eye. The second big challenge is in the genetics. How to get the protein absence genes to express in adult human neurons. So the question we're interested in, well, can we control protein activity without genetic modification? So this is where my background come in. So I was trained in mechanical engineering, but with a major focus in bioengineering, especially bioheat and mass transfer. There's a technique, a method called hypothermia or tissue hypothermia, so which basically increases the tissue temperature locally to ablate or to destroy cancer cells. This can be done by using a laser. For example, you can insert the laser into the brain and ablate the brain cancer cells. This can be done by, for example, a magnetic approach where you inject magnetic particles into the brain tumor and remotely excite the magnetic particles to heat and destroy cancer cells. So what we would like to do is to explore whether we can push the hypothermia from the tissue level to a molecular level. So to do this, there are a couple of important factors. So just like if you put some dye in water, it'll diffuse over time. So heat would also diffuse over time. So for tissue hypothermia to heat tissue, we're talking about typically increase the temperature by 5 or 10 degrees, and to treat a tissue that's on the order of a millimeter or centimeter size. If we go down to a molecular level at the nanometer size, then because the heat diffuses over time, so we really have to do 2 things. So one, because the size is very small, so we have to keep the time very brief as well. So we have to significantly reduce the time duration of the heating so we can localize the heating at a very small scale. Because we have to use very short duration, that means that, if you think about the protein denaturation, changing the protein activity as a chemical reaction, so we have to really increase the temperature significantly to increase the reaction rate in order to be able to denature or change the protein activity within a very short amount of time, a very confined and nanoscale size. So in this case, we're really talking about increasing the temperature of the molecule by hundreds of degrees and for a very brief period of time in terms of nanoseconds. So does this work? So we all, everybody probably knows how to cook protein, denature protein, right? So you've probably done this in the morning. And the question we would like to ask is whether we can cook or inactivate or denature individual proteins in live cells without killing the cell. So that's what we set out to do a few years ago. So there are many proteins of interest. We picked one called PAR2, protease activated receptor 2. And this is a protein that's very important in chronic pain. So cleavage of this part of the protein would lead to what's so-called a pain sensitization. So that means that even a slight perturbation, slight touch would cause significant pain. So first of all, we we set out to make a nanoparticle, gold nanoparticle that are conjugated with the antibody to target PAR2. And we see that when we target the PAR2, we see the nanoparticle localization on the cell membrane. If we don't target, for example, using gold particle with a PEG, a polyethylene glycol coating, we see that there's no significant cell accumulation on the surface, on the cell membrane. So then we set out to test whether this works. So there are a number of groups that I won't get into significant details, but the group of interest is that when we target the PAR2 molecule and apply a very short nanosecond laser, we see that there's a dose-dependent decrease in PAR2 activity. So you can see that around nanomolar range, we can significantly increase the PAR2 activity by over around 80%, while all the other groups seem to remain constant. So there are a few key questions in this. First of all, and which is also most obvious, is does the molecular heating or molecular hypothermia injure the cells? By looking at a number of assays, including the cell membrane integrity using PI and also cell proliferation, we can see that there's not any significant cell injury. Another question is because we use an optical approach, so we can, for example, shine light on certain areas of certain cells while blocking the light in other cells. We can see that only on the cells that we shine light, we can see the activity change, while on the other side, if we block the light, the cell activity maintains. So it has a spatial resolution at the light optical resolution. Now, this is different from molecular specificity because let me elaborate on this a little bit. There are many different protein receptors on the cell. There's PAR2, of course, there's many other types. So thus inactivation of PAR2 leads to changes in other protein activity. So for example, we pick this other receptor called SST receptor. And we see that while there's changes in PAR2 activity, there's no significant change in the SST receptor. Now, this is different from what I just talked to you in terms of you can use light to, for example, to shine light on certain cells, but not other cells. This is optical resolution. This molecular specificity is really beyond the optical diffraction limit. Because the protein size is only a few nanometers. So really here we're really relying on the nanoparticle to target the membrane receptors and to achieve this molecular specificity. So what happens to the PAR2 activity over time? So we track we culture the cell over time, and we find that there's an initial decrease in the PAR2 activity. And over time, over the course of 6 to 8 hours, the activity gradually recovers. So what we think here is happening is that once the protein molecule gets denatured, it's not going to recover by itself. Rather, this is a cellular process where the membrane receptors are being recycled, and the cell would synthesize and make new proteins, new part 2, and this would lead to the recovery of the part 2 activity. So this is a transient photoinactivation process. So and the next question we're interested in is how fast is this protein activation? There's a lot of work, an effort to try to measure the protein denaturation or inactivation at the cellular and the tissue level. Very less is known at the molecular level. So there, one of the models that are used to characterize the protein inactivation is the Arrhenius model. I'm not going to get into too much details of this equation, but just to say that the rate, the reaction rate, if you write it in the natural log of the reaction rate, is a linear function of 1 over t. Okay, this is what the model predicts. There has been some speculation on whether this is truly linear over a large temperature range, but nobody has been able to prove this. So we surveyed the literature by looking at both experiments and simulations. And the simulations are mainly molecular dynamic simulations that can increase the temperature dramatically and can only calculate at a very short time duration. The experiments are the solid dots. They're typically done, for example, by laser temperature jump and bulk heating experiments. If we overlay our experimental data on this graph, we see that it somehow overlays with the molecular dynamics simulation. Of course, there's some, not all of them. If we look at the trend, we can see that at low temperature, because this is 1 over t, on the right side, the curve looks linear, but at the left side, it looks like the curve is saturated. So this leads us to adapt and propose what's called a reaction diffusion model, where in a low temperature range, the temperature increase will significantly increase the reaction rate. So this is in the reaction limited regime. At high temperature, the further increase the temperature doesn't lead to increase in reaction rate. This is saturated, so this is in the diffusion limited regime. And there's a transition zone in between. So the last question we're interested in, what we've looked at, is how can we achieve, under what conditions can we achieve this nanoscale localized heating? Because any heating overlap would need to lead to non-specific protein activation. For example, if we increase with the same nanoparticle density, if we increase the duration of the heating, we can see that the temperature start to overlap, the heating start to overlap, and you get this non-localized, delocalized protein activation. If we look at this systematically, there are two important parameters. One is the laser duration, heating duration. The other one is the density or area density of the nanoparticles. You can see that we can build basically a surface where below the surface there's no overlap. Above that, you can have a significant overlap. If we look at one of the laser intensity, you can see that there's kind of a green zone where there's no temperature overlap. And above that, above a certain threshold, you can get significant temperature and heating overlap. And this seems to correlate quite well with the fact that if you don't have a significant temperature overlap, there's a very little amount of protein denaturation. But above that, you're going to get a catastrophic increase in the protein denaturation. So if you're somewhere over here, what we're doing is basically heating and cooking the entire cell. If we're in this range, we're doing a targeted and localized heating on the targeted molecules. And to be quantitative, this is roughly at about 10 nanoparticles or 10 receptors per micrometer square for a nanosecond laser. So to summarize the first part, I've described a new technique called molecular heating or molecular hypothermia to obtain transient photoactivation of protein activity without genetic modification. We've demonstrated cell membrane protein called part 2, and looked into the kinetics of protein activation at over a large temperature range, including high temperatures, and looked at on what conditions can we get nanoscale localized heating. As a next step, we're looking into how to quantify the nanoparticle targeting, how many receptors are on the cells, how much we're targeting, how much are we inactivating, and how that leads to cell signaling changes. So that summarized the first story. So basically, molecular hypothermia is a new tool to control protein activity without genetic modification. So now I'm going to switch gears to talk about the second part, which is how we can use nanoparticle, nano tools to understand basic and important questions in the brain. So in the nervous system or in the brain, there are two important methods that neurons communicate information. So one is by what's so-called fast amino acid transmission or synaptic transmission, where molecules are released into the synaptic cleft and diffuses a very short distance within 10 nanometers and transmits the signal to the next neuron. This is the main mechanism of the wire transmission or synaptic transmission in the nervous system, and it's very fast and travel long distance. The other mechanism is what's so-called a volume transmission. What that means is basically the cell releases molecules like peptides or neuropeptides into the extracellular space that diffuse locally and bind to other cells through what's also called the G protein coupled receptors. These are important to regulate brain state, to modify neural activity, and control vascular tone. So they play a very important role in the brain, and they travel a long distance, hundreds of micrometers to millimeter. So this is kind of like a biological version of engineering. So we don't really know how long they travel. So this is a kind of like a rough estimate. So the question we're interested here is, how do neuromodulators diffuse and act in the brain? And if we look at this curve again, look at the scales in the brain. So we're looking at molecules released from the vesicles. And if there's typically not just one vesicle release molecules, there's a lot of them. And once they get released, they would diffuse and to affect a local circuit. There's some speculation on whether there's some flow or not, and this is an area of significant debate. So this question is not only important for basic neuroscience, but also has a translational impact, because lots of drug delivery you would rely on the diffusion of molecules in the brain. So we look into this problem through a sort of three aspects. To give an overview of this problem, let's look at three aspects in terms of neuromodulator, diffusion, and the function. So first of all, the structure. The molecules, once they are released into the extracellular space, they would have to diffuse through the extracellular space. Now you can look at the extracellular space using imaging techniques, for example, cryo-EM, which I think Minnesota has a very nice facility. And you can also use there's also increased interest using super resolution imaging techniques, so optical techniques to image at the nanoscale resolution, how the organization, morphology look like for the brain cells and processes in extracellular space. So we can start to get an idea about how the extracellular space look like, in the brain. How about the property? How do molecules diffuse? Well, from the engineering point of view, we can really model the extracellular space or the brain as a porous medium. We can write down equations in terms of transport, and there are a couple of important parameters. So one is the volume fraction. So this is the percentage of the volume of the extracellular space in the brain. So this is about 15 to 20%, around 20%. And the second one is the tortuosity. So this basically describes how much the diffusion is slowed down in the brain compared with water. So as you can imagine, for a molecule to diffuse through the tortuous space, and there's some confinement and there's some obstruction and so on, so it'll be slower than the diffusion in water. So these are the two important parameters. And there have been several methods to measure the extracellular space diffusion property. There are a few of them, and I'll go through briefly several of them that are widely used. So the first one is the method called ITI, or real-time ontophorensis. So this method is quite straightforward conceptually. You basically inject this TMA, the tetramethyl ammonium, into one electrode, and you measure it at another electrode at a small distance. This is a quantitative measurement. It gives both the volume fraction and tortuosity, but it can only do this one molecule, or very limited molecules. The second method is an optical method. It's called integrative optical imaging, or IOI. And the concept is also very straightforward. So you basically use a pressure injector to micro-inject a small volume of dye into the tissue, and you would just sit and watch and image how the dye diffuse. So because it uses pressure injection, so the volume injection volume varies significantly. That depends on the tissue. So you can only get the tortuosity. However, you can do a lot of molecules as long as it can be labeled by dye. And this is mostly done under one photon imaging. The last method is more recent with the development of super resolution imaging. This uses a nanotube or nanoparticle nanotube that can be loaded into the brain, and it's fluorescent, so we would just image and track the movement of individual nanotubes. So in that way, you can basically draw a picture as the nanotube moves. It draws a picture of how the extracellular space looks like, and you can also start to probe the sort of the viscosity or diffusion properties in the brain. So in summary, so the diffusion property can be measured using either the optical technique or and also the single particle tracking gives an idea of both the structure and the diffusion property in the brain. However, none of these techniques can have tried or can measure a real neurotransmitter or neuromodulator. So we still know very little about this. So I would spend, after my PhD, I spent some time at Joe Zaszinski's lab at University of Minnesota. Joe pioneered an approach where he loads different molecules of dyes into a liposome and conjugate gold particle on the liposome. And with a short laser pulse excitation, you can release the basically packaged molecules from the liposome very conveniently. So this becomes a very convenient and nice platform to load a number of different hydrophilic molecules in the liposome, and use a laser pulse, you can release this very conveniently. So is there a question, or should we address it later?
take questions at the end of your talk, so please proceed and then we'll take all the questions later.
Okay. So this method basically uses laser excitation of nanoparticle that disrupts the nanovesicle. And another advantage of this technique is that once you package the molecules inside, it will actually protect the molecule from degradation, because a lot of the molecules of interest, for example, peptides, once you inject to the brain, it'll be gone after a couple of minutes. So it's not very useful to, unless you can protect it from degradation, and then with laser excitation, we can study it. Okay, so how does this look like? So we made a large liposome, or GUV, a giant unit, a giant liposome, and once we excite it with a laser, you can see that the dye gets released, and we can kind of track we can image the release of dyes. So over the years, we have looked into different molecules to kind of show that this is really a versatile technique, that you can use this technique for a lot of different molecules, and not only dyes, but also molecules that act as a function, second messengers, IP3, and also different neurotransmitters and peptides. For example, in this case, we loaded this IP3 molecule and let them taken up by the cell, and we can trigger calcium activity in the cell. So we spent a lot of time trying to understand how this works, both in terms of the optical property, how much here we coat the liposome with the nanoparticles on the surface. So how much gold we coat would affect the optical property. With the laser excitation, especially the short laser excitation on the order of a picosecond or femtosecond, this will lead to changes in the nanoparticle morphology, and also how exactly are we poking holes on the liposome to release the molecules. So another area we looked into is how can we make increase the photosensitivity. So we worked with a group at Switzerland, Zambos group, where they have this kind of weird non-spherical particle. So long story short, when we coat the gold on the particle on this non-spherical vesicle, and we can this vesicle allows using much less laser energy to release molecules because of the unique structure. And this further allows us to look into deep brain regions. For example, in a mouse brain at 2 millimeter, you can release from both the spherical and non-spherical vesicle. However, at deeper distance, deeper regions, you can only release molecules from this non-spherical nanovesicle, because there's a lot less light when you go deeper, if you shine a light from the top. So this platform would allow us to do a lot of things. For example, if we can package the molecules inside of the nanovesicle and photo-release them in the brain, and be able to image and track the activity, the diffusion of the molecules. So to test, as a proof of concept, to test it, we first start with a dye molecule, and we can see that upon the laser excitation, there's an increase in fluorescence, meaning the dye gets released and it diffuses to the surrounding areas. And we can then analyze the diffusion properties. So this is looking at the intensity of dye at different distances from the center. By tracking the peak of the fluorescent intensity, we can analyze the time it takes for the dye to diffuse at to different distances, and couple this with the mathematics and equations. We can start to analyze, for example, the diffusion, the effective diffusion coefficient in the brain, and also the tortuosity in the brain. So this 1.7, the tortuosity matches quite well with what had been measured before. So this demonstrates that this platform can be used in live brain to measure, to probe molecular diffusion. In order to measure or image the diffusion action of neuropeptides or neurotransmitters, or neuromodulators, we need some sensors, because the molecules itself is not fluorescent. So one of the sensors is called sniffers, so cell-based neurotransmitter fluorescent engineered reporters. So in short, what this is that it's a HEK cell that are engineered to express a neuropeptide receptor, and once the peptide binds to the receptor, it leads to a series of signaling that leads to finally a fluorescent signal that can be detected. So this is a fluorescent cell-based sensor for neuropeptide. And the nice thing about the sensor is that it has nanomolar sensitivity. There are many efforts to do this, but this is one of the only one sensor that can achieve the nanomolar sensitivity. And you can use this in vivo to study, when the animal behavior, it leads to release of a certain neurotransmitters or neuropeptides in live brain, in live animals. So we couple these two technologies together. As a proof of concept, we co-implanted the cell, the sensor, and the nanovesicle. With the laser excitation, you can see that if we increase the laser power or give it more pulses, there's more response from the sensor. And the signal is specific from the release of the peptide, because if we don't package the peptide, then we don't see a response. To really probe at the neuropeptide diffusion in the brain, we designed an experiment where we have two implants. So the first implant, or we call the core implant, has both the sensor and the nanovesicle. And a second implant is some distance away that only has the sensor. So if we release molecules from the first implant, then it would diffuse. Of course, it would diffuse in all sorts of directions, but at some distance away, you would be able to measure the diffusion distance. So this is what we did. For example, in this case, there are 3 implants. So this is the core that has the sensor and the nanovesicle. These are the 2 satellite sites that only has the sensor. And you can see that there's a brief time delay between when the sensor signal reaches the peak. And if you stimulate at the satellite locations where there's no nanovesicle, then you don't get a response. So if we analyze this data across a number of different distances, this is a lot of mouse experiments, because you can only implant a certain number of sensors in one mouse brain. So we see that it generally, with increase in distance, there's the time for the sensor to sense the signal increases, and beyond the 250 micron, or for example at a 280 micron distance, we don't measure a response no matter what we do. So this may indicate that this beyond a distance, there's just not enough molecules that can diffuse over that distance, or the molecules get sufficiently diluted. So when we couple with this with the model, and we can start to look into quantitatively how the neuropeptides diffuse in the brain. For example, depends on the parameters that we model. There are certain maximum distance that the molecules can diffuse while reaching a critical concentration. And the diffusion coefficient is approximately similar to that of a similar sized dextran molecule. So in summary, this allows us to really make quantitative measurement of this kind of like a Wi-Fi range. So how far can you receive the signal from the neuromodulators? and be able to characterize the volume, transmission volume. So this is a point measurement because you have to implant the cells to discrete locations. So it's not very scalable, but it allows us to do certain measurements. Then as a next step, we're looking into this genetically encoded sensors that can measure, different molecules. For example, this is a very recent development in terms of sensors for neurotransmitters and neuropeptides. This is a dopamine sensor. There's start to have some peptide sensors coming online, so we're still in the process of characterizing them. So in summary, I've told you how to use the nanovesicle tools to image or to characterize the neuromodulator diffusion in the brain, to look at this fundamental processes in the brain. Next, I will transition to the last topic. The last story is how to cross the blood-brain barrier. So the blood-brain barrier is an important structure in the brain. So if we look at if we zoom in and look at the brain, if we zoom in, there's a lot of very dense vascular network in the brain. So the closest the vessels between each other is on the order of a couple hundred microns. And if we look at one of the smallest elements, which is a capillary, there's this structure called the blood-brain barrier, which basically composed of, this is the endothelial cells that are connected together by a tight junction. So this, you can remember this name, tight junction. There are other structures, for example, a basement membrane, there's a parasite, and there's a astrocyte infeed, and so on. So this forms this very tightly regulated blood-brain barrier that provides an optimal environment for the brain, but also it's a big challenge for drug penetration. So there has been several methods that developed to open the blood-brain barrier. So one of them is by using mannitol, so to osmotically shock the blood vessel. This obviously has a high neurotoxicity, and it also works on the whole entire brain. Opens the blood-brain barrier in the entire brain. Another very promising method is using microbubble and ultrasound. So the method works like the following. So you make micron-sized gas bubbles and inject it to the blood circulation. And you can use ultrasound to remotely excite the microbubbles to that'll oscillate, that'll expand and contract, that'll stretch the vessel to open the blood-brain barrier. This method has certain advantages in terms because the ultrasound can penetrate deeper in the brain, and there's a lot of interest to move this to the clinic. However, one important aspect is you don't want this opening the blood-brain barrier to do damage to the brain activity. So you probably have heard of this FMI, functional magnetic resonance imaging. The University of Minnesota is also a leader in this area. So basically, it measures what's called a bold signal, so blood oxygen level dependent signal. And what this is that the brain activity, if there's an increase in brain activity, that would lead to increased neuron firing, neuron activity, and the neurons would release molecules to tell the blood vessel that, okay, I need more oxygen, nutrients, and the blood vessel would dilate. So you can see this oscillation, the dilation, constriction of the vessel. And the dilation of the vessels would reduce the fluid resistance. The more blood flow goes to the area, and that increases the oxygen level that can be detected by the magnetic signal. So this is how the physiology and how the fMRI works. There has been several studies that have shown that recently this ultrasound blood-brain barrier opening can lead to decreases the neurovascular response. So it kind of attenuates or in certain cases diminishes the neurovascular response. And also it causes disrupts the functional connectivity between different brain regions. For example, in this case, you open the blood-brain barrier, the connectivity between this region and other regions is significantly reduced. So the question we're interested in, we would like to ask is, are there other ways to safely to get through the blood-brain barrier? So one hypothesis we came up a few years ago is, can we gently push or gently perturb the blood-brain barrier by using a laser excitation of vascular targeted nanoparticles? Let me explain how this works. So we hypothesized that we can use nanoparticles to target or label the tight junction using after injecting nanoparticles systemically, and then we would apply a laser pulse to excite the nanoparticles to gently exert forces locally, very locally. And if we use a short laser pulse, this is mainly a mechanical effect. So they would generate pressure locally to perturb the tight junction or the blood-brain barrier. So does this work? Well, we first check the nanoparticle biodistribution. So as expected, then if you inject a nanoparticle to the blood circulation, a lot of them would go to the liver and spleen. But we do have increased accumulation. Let me back off a little bit. If we use the tight junction targeting nanoparticle versus a passive, polyethylene glycol coated nanoparticle, we see that the biodistribution is different. So there's a less accumulation in the with the tight junction targeting. There's a less accumulation in the spleen. There's more 4 times more accumulation in the brain, and there's also other changes in other organs. And we further confirm that the nanoparticles are indeed labeled on the blood vessel using either using silver staining or looking at the electron microscopy. We can find some nanoparticles that are sitting on the tight junction. So when we apply a laser pulse, we can see that in this case a blue dye would leak into the brain, and this is this opening of the blood-brain barrier is dependent on the laser energy and time. So basically, with high laser energy, we open it a larger area, and also it stays longer. Let me go into this into a little more details. So if we inject different dyes, for example, this easy link biotin is a small molecule, about 500-600 dalton. This viscodextrin is a large molecule, it's several kilodalton. We can see that initially, 2 minutes or 1 hour, you see both dyes get into the brain, right? So the red and green overlay, right? However, at 6 hours, we only see this red signal, or we only see this small dye gets into the brain, and at 24 hours, none of them gets into the brain. So blood-brain barrier recovers at that point. So by doing this experiment, this would inform the potential mechanism of how the BBB permeability increases. Because there is a size-dependent leakage at this time point, we think that this may be a paracellular diffusion, because the diffusion through the paracellular tight junction is size-dependent. Small molecules can pass through, the large molecules can't. So this suggests that. Versus if it's a transcellular pathway, it would take up both the small and large molecules and get into the brain. We looked into this further by injecting a heavy metal, in this case called a lanthanum injection. We see that if you don't apply the laser, the lanthanum would partially fill this tight junction. This tight junction goes all the way here to the basement membrane and so on. When you apply the laser, we see that the lanthanum would fill the entire tight junction, and it would go further to diffuse into the basement membrane and the interstitial space in the brain. So this strongly suggests a paracellular diffusion pathway in terms of how we modulate the BBB permeability. Does this do any damage to the brain? So we looked at a few things. So first of all, we looked at brain vasculature by labeling the blood vessel with lectin, and also looking at CD31. There's no major changes in both. How about the neurons? So we did a lot of immunostaining looking at neurons and also the axons, and the same, pretty much the same. What's more convincing is this Golgi staining, which is very classic tool in neuroscience, where you see that these two sides look morphology. In terms of morphology, look very similar, and they have this structures. They're very similar to each other. So we think there's not a lot of neuron damage in the brain. Critically, are we doing any changes to the neurovascular response or the viscodynamics? So we performed a multiphoton in vivo imaging, looking at the blood vessels in the brain before and after the BBB modulation. We see that in the arterioles, you would expect this kind of oscillation, constriction, dilation in the vessel periodically. And after the BBB modulation, you see that this signal persists. And there's no, in the venules, there's no vessel motion. And if we do a Fourier transform, we see that this vessel motion at about 0.1 hertz persists throughout the during the BBB modulation. So we think that this is this does not impact or does not disrupt the neurovascular response in the brain. How about the drug delivery or antibody and gene delivery? So we show that we observe that after we apply the laser, modulate the BBB permeability, we see increased uptake of the IgG, both the endogenous mouse IgG and also injected exogenous human IgG. If we administer gene virus vectors, for example, in this case, AAV, adeno-associated virus, into the systemic circulation, we see that this you can see, a lot of neurons are transfected, shown up green here, suggesting that there are, it's possible to do gene therapy and antibody therapy for different brain diseases using this method. So in summary, I've shown you a new way to modulate the blood-brain barrier using light stimulation, in this case, a picosecond laser stimulation of molecular targeted nanoparticles. This acts mainly through the paracellular diffusion, where it diffuses through the compromised or the tight junction. It does not disrupt the vasodynamics, and allows antibody and gene delivery. So, to summarize the talk, I've talked to you about 3 stories. The first one being using new to molecular heating, molecular hypothermia to control protein activity without genetic modification. The second being using nanovesicles to package and release neuromodulators and image neuromodulated diffusion in the brain. And last being using laser excitation of nanoparticles to cross the blood-brain barrier without disrupting the neurovascular response. So with that, I would like to stop here and answer any questions.